Namaskar, warm welcome to this wonderful session. This is part 7 of chapter 8. By now we have discussed 6 parts of this chapter. In this chapter we have to discuss gram staining regarding that is prokaryotic cell bacteria. Then we have to differentiate what we mean by gram positive and gram negative bacteria, their differentiation. And then we have to see differentiation in prokaryotic and eukaryotic cell. As you know that this is the last part of this chapter, number 8. In last part, we have to discuss NCERT exemplar given in the end of this chapter, and then we have to see previous year examinations questions. Some of the questions we have to discuss. This is part 7, last part, chapter 8, the cell unit of life. In this part, first we have to discuss gram staining, a very common term related to the prokaryotic cell. Frequently we use gram positive, gram negative bacteria or prokaryotic cell. So we must be familiar about these terms. That is what in by gram staining and what are the differences in gram positive and gram negative bacteria. See children, gram staining that was developed by that is Christian Gram, and that's why just to give respect to him, this process or staining is known as gram staining. This is a special type of technique which is used to classify bacteria into two groups, that is gram negative and that is gram positive. The bacteria, we have to see detail of this, that bacteria that take up gram stain, which retain gram stain known as gram positive, and in which the stain will be washed out, they do not retain it, these bacteria are known as gram negative one. So Gram is the name of scientist, Christian Gram, who has developed this technique to classify bacteria into gram positive and gram negative. You see a flow chart I had given how to differentiate and what's the procedure. What we have to do, first step, you see this is bacterial smear on a slide, that is we will put some that is bacteria, like this is curd bacteria, lactobacillus bacteria this is. We will dry. A thin film of the bacteria will be there on this slide. It is called that is bacterial smear. That will be air dried. Okay. Now on this we will put with the help of dropper, we'll put the stain. Stain here is crystal violet. Crystal violet or also known as gentian violet. This stain will be put here. What will be the result now? That is after this staining will be for about 30 seconds. That is all they will become dark in color. They will take all bacteria, they will be colored. Their color will be that is violet color. Bacteria get stains, they will become bluish purple or violet. After this we have to wash with water. We have to put water here and we have to wash. Okay. After washing, that iodine staining, is it 0.5 stain of iodine works as mordant. We have to that is apply now, that is iodine staining. Okay. After this what will happen? Formation of crystal violet iodine complex will be formed here in this, and all they will become, that is this color will be established, that will become more darker one. Now the important step here is, after this, that slide will be treated with alcohol. When we have washed it with alcohol, so we will get two groups. Some of the bacteria, they will remain even after washing with alcohol. They will retain that is this stain, the color will be retained here. Some bacteria retain this crystal violet color, and these bacteria will be known as gram positive one. While the other one, that is this stain will be washed here in this case, that is the color is retained even at present. These are colored, while in this case the stain will be washed and they will become that is colorless. These stained bacteria, that is counter staining with safranin. Remember that is they have taken this crystal violet color, but these bacteria, if you want to see them, we have to color with this safranin. Safranin is also one of the stain, and then they will become that is pink color. The bacteria which will take this safranin, which will become that is pink in color, known as gram negative. But the important thing is that is in which the stain will be washed out after treating it with alcohol, known as that is gram negative, and the stain is retained, that is known as gram-positive one. Is it clear? Very important flow chart. So by now we have divided bacteria into two groups, that is gram-positive and gram-negative. Now you have to see what's the reason that some of the bacteria they have become gram-positive and others these are gram-negative one. We have to differentiate, that is what is the reason, that is some have retained the color, that is crystal violet, and the other this crystal violet color is that is being washed away. We have to see the differences between gram-positive and gram-negative bacteria. Gram-positive, as I told you, first point, that the bacteria remain colored blue or purple with gram staining even after washing with alcohol. Very important point, that is here they do not retain color, they have become colorless when have been washed with alcohol. Okay. Outer membrane is absent. You see children that this is the bacteria we have here, that is outer membrane is absent, while in these bacteria outside this cell wall the membrane is present, which is known as that is outer membrane. Cell wall is this, see the cell wall is thick. If I am going to make cell wall of this bacteria, cell wall is thicker, it may be 20 to 80 nanometer thick, cell wall is thicker, while here the cell wall is that is comparatively thinner, about 8 to that is 12 nanometer. An important point here is that here the cell wall is thin, but even then it is having that is two layers, outer layers and the inner layer, but here such layer is absent. Cell wall is smooth, as you are seeing here, wall is wavy and come in contact with plasma lemma at so many that it is wavy cell wall one is. Very important point this one is, you know that is in bacterial cell wall murin or peptidoglycan. What is this? Urine or that is peptido, that is glycan is present. Peptidoglycan is found in bacterial cell wall, but here in this case the content of that is that is this peptidoglycan or murin is very high, 7 to 80%, while the murin content here is less, it is 10 to 20% only. This additional information we have, basal body of the flagellum contains two rings S and M. You can see little detail of this, that this is the cell wall, let this is the plasma membrane we have, this is cell wall, this one is that is cell wall, let this one is that is plasma membrane. Here you know that is the flagellum arises, this is the that is inner part, that is basal body, this is the hook I told you earlier, and this is the that is flagellum we have. This flagellum, you see this basal body in case of gram-positive bacteria, it has only two rings, and these two rings are S and M. One ring is here, right? One ring and two rings. This is S and M. Only the two rings are present in plasma membrane. While in this case, gram-negative bacteria, just you can remember for reference, that is this is the cell wall, right? This one is the plasma membrane. Let this is the that is basal body of the that is flagellum. That this is the hook we have here, right? This is hook, and this is the that is flagellum. This one is the flagellum here. Here the flagellum is having four rings. The two rings are present in that is plasma membrane, and the two rings are present in cell wall. The rings present in cell wall, these are known as L and P, and here the rings present here, these are known as that is S and M. Is it? In this case, only the two rings are here. These two rings are, these are S and M, while here we have the four rings. This sometimes may come in examination. It is having teichoic acid. Teichoic acid is present in gram-positive. It is absent here. Teichoic acid is absent. That is, a few pathogenic bacteria belong to gram. Most of the non-pathogenic bacteria belong to this group. Only a few forms are which are pathogenic one. Most of these are non-pathogenic one because they do not make capsule also. Here that is most of these are that is pathogenic one. For example, like this is Salmonella, like this is Vibrio. These are pathogenic bacteria. Is it clear? Okay, come to the next now. We have to say now many more times in chapter we have discussed about the types of cell. One is prokaryotic one. You know prokaryotic one. Here this is the nuclear material which doesn't have any And yes, it doesn't have any nuclear membrane. Here we do not have any cytoplasmic organelle. Only what we have, that is 70S. We have, that is ribosome we have here. Many more types you have seen here. While in case of eukaryotic cell, children, in the beginning also I told you about eukaryotic cell. It is having a well-defined nucleus which is having this, that is nuclear membrane, double nuclear membrane is there, where this nuclear content is there, and it is having cytoplasmic organelle. Can you recall? After that we have seen one by one. It is having endoplasmic reticulum, it is having Golgi bodies, it is having what? Lysosome, it is having, that is vacuole. Do you remember? It is having what? That is chloroplast in plant cell. It is having, that is mitochondria. Can you recall few more? It is having, that is cilia, or that is flagella, is it? And it is also having what? That is centriole. These things in detail I told you, but we have to summarize one by one. Prokaryotic cell, the things which have not been covered so far, that is nucleoid. Start from here. Nucleoid, not membrane bounded. This structure is known as nucleoid. Here the nucleus, this one is membrane bounded one. No membrane bounded organelle I told you here. Here the membrane bounded organelles are present. That this one is the mitochondria. That this one is the Golgi body here. These are present here. Mainly single chromosome is there, but some bacteria may have two. Single chromosome. Here that is more than one chromosome is present in, that is eukaryotic cell. You know, in case of human being, we have, that is 46 number of chromosome. No histone protein in chromosome, very important protein. That is point. No histone. Here DNA, that has, it is covered with, that is histone protein is present. DNA transcription messenger RNA translation, that is both occurs simultaneously. You have to study, but you have to see the detail of this. That is here. Here in case of, you can see this point here. That is here. That is, this is the DNA which is present in cytoplasm. Here the messenger RNA will be formed. Process is known as transcription. Then from messenger RNA, the protein will be synthesized. So this process is known as formation of messenger RNA from DNA is known as transcription, and this process is known as translation. Both of these, they take place simultaneously in cytoplasm. Here what will be here? DNA transcription will take place in nucleus. Here that is transcription will be there. Messenger RNA will be synthesized here. Then this messenger RNA will be transferred to cytoplasm where protein synthesis will take place. Okay. That is, genome is haploid. Haploid genome, only one. Mainly diploid genome are present. That is many, especially in plant, they have even more than two, known as polyploidy, 3N, 4N, 5N polyploidy. DNA plasmids are present. You see that this is the main, that is this hereditary DNA, but some DNA is present in cytoplasm also, known as plasmid DNA, which perform different type of function. This is the DNA outside the chromosome. Plasmids are not found in eukaryotic cell. The compact genome, little repetitive DNA. You have to study in time to come what in by repetitive DNA. That is here, lot of non-coding and non-repetitive DNA is present. That is here you see, that is this one is, that is exon and intron. Here in this case, introns are absent. Introns, they have repetitive DNA, while in this case, that is exons, these are alternated with intron, that is exon and intron. Detail of this you have to see in time to come. No gene introns are present. No genes introns are absent. The genes they have, that is introns, single DNA replication origin. That is, this is the DNA, the circular DNA is there. That is, here we have single replication origin. Origin is only one, where the DNA replication will start. Here in this case we have multiple DNA, that is origin. Here ribosomal RNA sedimentation coefficient is low. You all know, very important point this is. That is, in this case we have 70S ribosome. Here we have, that is 80S ribosome. Smaller, that is ribosome. Larger, you see smaller ribosomal, that is this is, and larger RNA in cytosol, smaller, that is what you mean by this point. Here we have only 70S ribosome found in, that is cytoplasm. Here we have larger one, 80S we have in cytoplasm, and 70S we have in some organelle. Can you recall these organelle? That these are, that is like chloroplast, and let this one is, that is mitochondria. Okay. See some more important, that is vacuoles are there in this, but these are gas vacuoles, no membrane bounded vacuoles. Here, that is may have vacuoles, especially in plant cell. That this is the plant cell I'm going to make here. Then in this plant cell, this is the cell wall, you know, in case of, and here we have a large central vacuole is there, known as vacuole, and this membrane is known as, that is tonoplast. The membrane of the, that is vacuole is known as, it is called tonoplast. Okay. Plasma membrane, that is no receptor, you can remember it. You can, plasma membranes, these are with receptors, and it is having sterols and carbohydrate. Chemically complex cell wall made up of peptidoglycan. Here the cell wall, if is present, that will be of cellulose, and that is chitin, as in case of fungi. Detailed structure up I told you. You must be remembering that this is the bacteria, this is plasma membrane. Plasma membrane, it is covered with, that is what is cell wall, and cell wall is covered with, that is glycocalyx. All together what we used to call, that is known as cell, that is envelope. You must be remembering. So this cell envelope is found in case of bacteria prokaryotic cell, while such things we do not have in eukaryotic cell. That is glycocalyx is present. They have mucigenous sheath. All these things are absent in prokaryotic cell. Rarely microtubules are absent. I told you just now. That is, while we have the microtubules are present in eukaryotic cell. No cytoskeleton related to this. No cytoplasmic movement related to this. Here we have cytoplasmic movement, that is around this vacuole. The cytoplasm, that this is central vacuole, it used to rotate clockwise, or that is endoplasmic, anti-clockwise, known as cytoplasmic streaming. Cytoplasmic streaming, you know, is also known as, that is cyclosis. It is found only in case of eukaryotic cell. Okay. That is lysosome, no need to recall it, lysosome, peroxisome, because these are also cell organelles, membrane bounded structures, which are altogether absent in prokaryotic cell. That is, they may be present in eukaryotic cell. That is buoyancy, gas vacuoles are present. That is, as in case of, that is blue-green algae. This blue-green algae, it used to float on surface of water. It is because of, that is, gas vacuoles are present here. Most of these are strictly anaerobes, that is aerobic form, also facultative anaerobe. You know the meaning of this? Strictly anaerobes, most of these, that is, yes, what is this? Strictly anaerobes, that is, they will survive in absence of air, in absence of oxygen. They may be, that is aerobic, also facultative anaerobes. What do you mean by? If the oxygen or air is not available, that a few may survive in absence of air and oxygen, known as facultative anaerobes. Is it? Size you can see here, that is, this is 0.5 to 100 micrometer. The size will be this one, right? Some are able to do nitrogen fixation. Nitrogen fixation, you know, can be done only by prokaryotic cell, only by prokaryotic cell. That this is blue-green algae, and so many other examples we have. That is higher, that is eukaryotic cell. They cannot fix nitrogen, very important point. That's why we have to salute. We have to salute to this, that is tiny organism, prokaryotes. That is all nitrogen fixers belong to this category. The free nitrogen, atmospheric nitrogen, can be fixed only by prokaryotes. That is, they have short cell cycle division, that is 20 to 60 minutes long. That is, they have, that is 12 to 24 hours. They usually reproduce by, that is binary fission, may be horizontal gene transfer, known as conjugation. You have to study in detail, and I told you earlier, conjugation method. That is, two bacteria of different strains will come close, and then they will transfer the, that is gene. But even then, that is diploid condition is never achieved. You must be remembering conjugation is there. That type of sexual reproduction is known as primitive type of sexual reproduction. Here, because we have mitosis and meiosis, they do not have conjugation. Here we have alternation of generation in cells. There will be no alternation of generation. Is it right? Okay. Come to the next now. That is, beside this, we can recall some that is other important points. That is some other differences between that is prokaryotic cell. If you see that is size of the prokaryotic cell, these are usually larger as compared to that eukaryotic cell. These are usually larger size. These are that is smaller one. Is it? This is also one of the difference. These are usually that is primitive in nature. These are primitive, and these are comparatively these are that is advanced one. Okay. Some other points we can discuss. Come to the next now. As I told you, this is the last part of the chapter. As that is, we have to discuss what NCERT exemplar first, and then we will switch on to the next, that is previous year examinations question. NCERT exemplar. First, common characteristic feature of plant sieve tube and most of the mammalian ichthyosite. Do you remember that both they have nucleus when the cells were young, but during maturity they have lost the nucleus? That condition is known as anucleate condition. Anucleate 1. This is absence of nucleus when the cells are that is mature. In mature cell, nucleus will be absent. Select one which is not true for ribosome, made up of 2 units. They make polysomes. They may attach to messenger RNA. Definitely, site of this is messenger RNA. Ribosomes will be attached. Only then protein will be synthesized. Have no role in protein synthesis. That is not true for ribosome. Which of the following stain is not used for staining chromosome? Basic fuchsin, methylene green, carmine, all these are basic stains. Safranin is not used to stain this, that is chromosome. Stain used to visualize mitochondria, that is Janus green, also known as that is vital stain. This one is vital stain. Come to the next. Different cells have different sizes. Arrange the following cells in the ascending order of their size. Choose the correct option. You can write mycoplasma, human RBC, ostrich egg, and bacteria. There is no doubt mycoplasma is the that is smallest one. Ostrich egg is the largest one. No doubt that is first and the last. Human RBC and that is bacteria. Bacteria is definitely lesser than human RBC. The right answer will be first is mycoplasma, second is bacteria, third is human RBC, first is that is last one is ostrich egg. Which of the following feature is common to prokaryote and many eukaryotes? Only what? That is cell wall. Which is the following feature common to prokaryotes and many eukaryotes? Many eukaryotes means eukaryotes, you know, that is we have here that is protists. Is it? Most of the protists they have cell wall. Then this is that is fungi, and then this is kingdom. Plenty most of these they have eukaryotes. Out of 4 kingdom you can leave this kingdom, that is Animalia. This is similar to this, that is bacteria. Okay, cell wall is very important and interesting question. This is what is tonoplasm? I told you membrane which surrounds the vacuole, that is known as tonoplast. Previous year examinations question come to this, which appeared in NEET 14. Which structure performs the function of mitochondria in basis in bacteria? That is mesosome. That is mesosome. This is the invagination of plasma membrane. It is having mitochondrial enzymes. Select the correct matching in the following pairs. Yes, rough. Yes, you can see easily. Smooth. That is ER. It is oxidation of phospholipid. No doubt in this. I told you in advance. Detail of this. Nuclear envelope is derivative of rough endoplasmic reticulum. Which of the following structure is not found in prokaryotic cell? Plasma membrane is there. Ribosome is there. 70S ribosome. Mesosomes, I told you. The only thing which is absent, that is nuclear envelope. Come to this next. That is in 2015. Cell wall is absent. Nostoc, it is there because this is what nostoc, this is blue-green algae. Funaria, this even is that is bryophyte. Cell wall is present. Kingdom, that is plenty. Aspergillus, this is what? This is fungi, that is chitinous cell wall is present. But in Mycoplasma, it is the bacteria without cell wall. That's why its colony, it appears, that is fried egg-like. Fried egg-like. That's why this is also known as Zoker of plant kingdom. It keeps on changing its shape. Respiratory enzymes occurs in bacterium. Question very important. That is being repeated here. Plasma membrane. Following components provide sticky structure, that is glycocalyx in bacterial cell. Which of the following cell organ is responsible for extracting energy from? Everyone knows it, being a student of biology. Mitochondria known as powerhouse of cell. Ribosome is known as engine of cell. Protein synthesis. Lysosome is known as suicidal bag. Chloroplast is the site of, that is, photosynthesis. Which of the important site of formation of glycoprotein and glycolipids? Very important function of Golgi bodies, packing material, Golgi bodies. Which of the following statement about inclusion body is incorrect? These are involved in digestion of food. No, that is, digestion of food will be with the help of lysosome. Is it right? So this is part 7, chapter 8. Last part it was, and in that way we have finished chapter 8, cell, the unit of life. Thank you.